Okay guys, if you had trouble loading this electrophoresis simulation because of Flash, I'm just going to walk you through it here in the video so you can use this in place of that if you couldn't do it. It'll probably be a little bit more interesting if you actually do it yourself, but this will work to help you understand sort of the basics of electrophoresis. I guess I'll read what it's saying as well as click through what's supposed to be done. You're holding a small plastic tube with some clear liquid in it. You've been told that the liquid contains DNA strands of several different lengths. Your job's to figure out what those lengths are. How will you do it? If the DNA strands were as big as your shoelaces, you could sort them out by hand into groups and measure them. The DNA strands are molecules so tiny that you can't see them, even under most microscopes. Is there a way to sort and measure the DNA strands in your tube even though you can't see or touch them? There is. It's what's called electrophoresis. Scientists use gel electrophoresis whenever they need to sort DNA strands according to length. This technique is also useful for separating other types of molecules like proteins. How does this work? The gel is the filter that sorts the DNA strands. It's like a sponge made of jello with many small holes in it. We place DNA samples into the holes at one end of the gel. Electrophoresis is how we push the DNA strands through the gel filter. By adding an electrical current, we can make the DNA move. Short strands move through the holes in the gel more quickly than long strands. Over time, the shorter strands in the sample will move farther away from their starting point than the longer strands. DNA strands of the same length will move the same speed and end up grouped together. In this way, DNA strands in the sample sort themselves. Staining the sorted groups of DNA makes them visible to the naked eye. Although we can't see a single DNA strand, we can see large groups of stained DNA strands. These groups show up as bands in the gel. Now it's your turn to run a gel. That's scientist slang for gel electrophoresis. Follow along with the steps shown above. Here's what you'll need to make the gel: powdered agarose, buffer, a flask, a microwave, the gel mold, and the gel comb. Making electrophoresis gel is a lot like making jello, but you wouldn't want to eat this type of gel. Put a small amount of agarose into the flask. Agarose is a dried powder similar to gelatin, but it's made from seaweed. Add some liquid buffer to the flask. Buffer is a saltwater solution that will let electrical charges flow through the gel. We've loosely placed plastic wrap over the top of the flask to prevent the liquid from boiling over. Open the microwave by pressing the open button. Place the flask containing the buffer and agarose mixture inside of the microwave. Heat the mixture until the agarose melts into the buffer. We've removed the plastic wrap from the top of the flask. Pour the melted agarose mixture into the mold. Notice that the mold has tape on each end to hold the melted agarose. Place the comb into the gel at one end. The notches in the gel will hold it in place. Let the gel cool and solidify. This usually takes about half an hour, but we'll speed up the clock for you. As the gel cools, tiny holes will form in it. Carefully remove the comb, leaving empty wells for the DNA sample. Now it's time to set up the electrophoresis box. You'll need the gel you just made, an electrophoresis box, and another bottle of buffer. Pour the buffer into the electrophoresis box. Place the gel still in the mold into the electrophoresis box. We have removed the tape from the ends of the gel mold. The gel should be just barely submerged in the buffer. The buffer conducts the electrical current from one end of the gel to the other. It will also keep the gel from drying out during the experiment. You're ready to load the DNA sample into the gel. Here's what you need: loading buffer, the tube of DNA, the DNA size standard, a micropipetter, the electrophoresis box containing the buffer and gel and pipette tips. With a clean pipette tip, use the micropipetter to suck up some loading buffer. Then add it to the DNA sample. DNA samples are prepared in a clear liquid solution that would be hard to see if you tried to load it directly into a well. The loading buffer contains a dye that makes the sample easy to see. It's also slightly goopy. This makes the DNA sample thicker so that it will drop to the well instead of floating away. The DNA size standard already contains loaded buffer. Next, you'll use the micropipetter to transfer the DNA sample from the tube into the well of the gel. First, suck up some of the DNA sample into the pipette tip. Eject the DNA sample from the pipette into the first well of the gel. Nice job. In real life, loading the sample into the wells takes some practice, so don't just be disappointed if you miss your target the first few times. Use a clean pipette tip. Use the micropipetter to suck up some DNA size standard. Transfer the DNA size standard into the next empty well. The DNA size standard contains DNA strands of known lengths. Running it on the gel will give you a reference by which to estimate the lengths of the DNA strands in your sample. It's time to turn on the electricity and run your gel. When you turn on the power, the black will generate the negative charge. The red end will generate a positive charge. Together they will pass current through the gel. DNA has a negative charge. To move the DNA through the gel, you must put the black cord in the negative charge closest to the wells. Okay. Plug the black cord from the electrophoresis box into the matching outlet on the power supply. Click the location on the power supply where you want to plug the black cord. Now click the location of the power supply you want to plug the red cord. Turn on the power supply. Your gel is off and running. Let's look into the gel box and see what's happening. Check for tiny air bubbles coming out of the electrodes at both ends of the electrophoresis box. These bubbles are your proof current is running. Repelled by the negative charge, DNA moves through the gel toward the positive charge, the other end. Short DNA strands move through the holes in the gel more quickly than long strands. Over time, the shorter DNA strands will migrate farther from the starting point than the longer strands. We can't actually see the migrating DNA bands, but we can see the blue dye from the loading buffer as it migrates. You've finished running your gel. We've taken the gel mold with the gel in it out of the electrophoresis box. The time has come to analyze your sample and estimate the length of the DNA strands. First, you need to stain the DNA in your gel using DNA staining solution. The stain is a chemical called ethidium bromide, which binds to DNA and shows up under fluorescent light. Although we can't see single DNA strands, we can see large groups of DNA strands. These groups will show up as bands on the gel. Drag the gel out of the mold and put it into the DNA staining solution. Because ethidium bromide binds to DNA, it can damage DNA in your cells. If you stain a gel in real life, wear gloves and avoid direct contact with the staining solution. It takes about half an hour to stain the DNA in the gel. We'll speed up the clock for you. Remove the gel from the staining solution and place it on the UV ultraviolet light box. Turn on the UV box. UV light from the box can damage your eyes. If you do this in real life, be sure to wear protective goggles. Now you can determine the approximate length of the DNA strands in your sample. Compare the bands from the DNA sample with the bands of known length from the DNA size standard. Enter the estimated length in base pairs for each band in your DNA sample. Electrophoresis cannot tell us the exact DNA lengths of the DNA strands. It's just a good estimate. So give it your best guess based on the DNA size standard. Press forward when you're finished to check your answer. All of your estimations are correct. The estimated length and base pairs of each band in your DNA sample is based on how they line up with the bands of known length in the DNA size standard. The top band is approximately 6,000 base pairs. The second is approximately 3,500 base pairs. And the third is approximately 1,500 base pairs. Congratulations, you've just run your own DNA gel electrophoresis experiment. Press forward to start over. And that's your gel electrophoresis simulation.